So we'll wrap this up and then we'll go into the next chapter, which is gram-positive bacilli of medical relevance. Okay, let's get this all set up. Okay, so we were talking about, we first started with the staphylococcal bacteria, and then now we're focusing on the streptococcal bacteria. Strep throat is something we're all familiar with. Many of us have had it, and if we've had it, we're much more likely to have it and have it and have it over and over again. It's caused by, it's a streptococcal pharyngitis, and it looks like this. So it's really easy to diagnose. You just open up your mouth and look at the back of your throat, and if you see these white plaques on there, then it's a pretty good chance you have strep throat. Now, if it becomes there are certain systemic infections that are of streptococcal origin. Scarlet fever is from Streptococcus pyogenes, and it carries a prophage that, if you guys remember prophages, a phage is something that attacks a bacteria, a bacterial cell, and sometimes those prophages are co-opted to actually be beneficial for the bacteria. And so in this case, there's one that secretes this erythrogenic toxin, okay? And so that can lead to sequelae or long-term damage as a result. Septicemia can be a huge issue, right? Because once you go septic, that means your whole body is basically systemically infected. It's worse than bacteremia or viremia, right? This can also lead to pneumonia, and then what we call streptococcal toxic shock syndrome, which we talked about a little bit earlier. Now, if you have these streptococcal infections, one of the dangers is getting something like rheumatic fever, okay? And what happens is that when children in particular, but not just children, have some kind of streptococcal pharyngitis, but it's subclinical, meaning, it doesn't manifest with those big plaques on the back of the throat, or maybe just the children, aren't effectively communicating that they feel bad. That bacteria can reach the heart. And when it does, it can wreak havoc on the valves of the heart. Okay. And so this is typically why strep infections are treated, not to treat the strep throat, but to prevent it from going into the next phase of rheumatic fever. Okay. You can also have acute glomerulonephritis, where it affects your kidney, and you can have kidney failure, right? And so these are, again, long-term issues that happen when the infection goes from a localized to a more systemic manifestation. That was group A. With group B, this is going to be things like Streptococcus alligactiae, and that's present in the vagina, the pharynx, and the large intestine. It can be transferred to infants as they're being born through the birthing canal, and if so, that can cause pretty severe infection. Pneumonia, sepsis, meningitis, so inflammation of the meninges up, in your brain. So typically, women are screened for group B strep very commonly. when they're pregnant, and especially, in the last trimester, because if they are positive, they need to go on an antibiotic regime to get rid of that, so it's not passed on to the infant. Wound and skin infections can then lead to endocarditis again, so it can go systemically, reaching the heart, and can be very debilitating. Group D is Enterococcus faecalis. It's still a strep, even though it's not streptococcus. It is a streptococcus bacterium. Enterococcus faecium and Enterococcus durans. And these are oftentimes normal flora in the large intestine and not harmful, but they can be opportunistic. And so sometimes if they're in the urinary tract or if they're in a wound, then they can be deleterious. They're no longer just normal residents. So an example of a bacteria that is not harmful or harmful depending on the tissue that it's residing in. Group C and G, that is common in our pets, and oftentimes resides in the upper respiratory tract and can lead to pharyngitis. It can also, if it goes more systemically in a bacteremia-like manifestation, it can lead to glomerulonephritis, again, attacking the kidneys. We talked on Monday about alpha and beta hemolytic bacteria. So the beta hemolytic bacteria completely lyse the red blood cells, where when alpha, they lyse it partially. And we talked about, does anybody remember the enzymes that are used for that, respectively? Like, what do alpha hemolytic bacteria use? No, streptolysin was one, and peroxidase was another one, I believe. So I'll let you guys review that earlier in the slide deck. It might be helpful to know, the difference between alpha and beta hemolytic bacteria. There are a lot of members in this group. Streptococcus mutans, oralis, salivarius, those are going to be in the mouth. Sanguis, malari, and mitis. And so these are very common, again, in the oral cavities, gums, teeth, and nasopharynx, but also present in the genital tract and to some degree on your skin. Not particularly invasive, although, they do lead to cavities. But when you are having any kind of dental surgery, or particularly if you have heart disease or having surgery on your heart, then it's very important to treat with antibiotics so that these bacteria don't have the chance to go systemically and then cause endocarditis and things like that. So not particularly bad in general, but when we do have surgical procedures, then we have to make sure that these remain localized. Bacteremia, meningitis, infecting the abdominal region, tooth abscesses, that these are all going to be symptoms and signs of these alpha hemolytic streptococcal bacteria. And of course, the most detrimental manifestation is going to be endocarditis, because it attacks the heart lining and the valves. And you have to have a properly functioning heart to live, right? That's just obvious. So Again, if you have a pre-existing heart condition or having heart surgery, um, then you need to be treated with antibiotics prophylactically in order to prevent this from happening. And once they do colonize, they can form these biofilms, which biofilms are very hard to treat. And so once it gets to that point, it becomes a much more invasive therapy. Streptococcus mutans produce slime layers, yay, that adhere to your teeth. And so that's the plaque on your teeth that builds up over time, especially if you don't floss. And then you go to the dentist and they have to scrape all that stuff off. That is from Streptococcus mutans. So the next time you go to your routine dental cleaning, you can impress the hygienist when she's cleaning off the plaque by saying, yeah, I've got a lot of streptococcal mutans, I guess, build up. Or maybe you can say, maybe she said, oh, or he says, there's not much plaque on your teeth. And you can say, yeah, not much streptococcus mutans. And yeah, you probably aren't going to do that because that would make you sound like a huge nerd. But it's up to you. Maybe you're proud to be a nerd. I am. But anyway, so again, pre-existing heart conditions, you're going to have prophylactic antibiotic treatment, okay, to prevent these pretty much harmless, for the most part. Again, they break down your teeth, but they're not going to really lead to a systemic infection unless you're being operated on. Streptococcus pneumoniae leads to pneumonia. And in fact, it's the primary cause of bacterial induced pneumonia. So pneumonia can come from viral infection, can come from bacterial infection, can come from fungal infection. But when we're talking about bacterial induced pneumonia, it's primarily from Streptococcus pneumoniae. And so these are going to be cells that are arranged in pairs or short chains. So they're going to be diplococci or streptococci. And they have the ability to form capsules on the outside to protect them. And that's going to be their major virulence factor. Okay. Fun fact, interestingly, there's been 90 different capsule types identified. So not all capsules are the same, right? They're slightly different in what types of surface proteins and antigens they have present on them. And so what happens is they can go into the lungs and start replicating, and that's what leads to the pneumonia. Or they can travel from the upper respiratory system through your eustachian tube that connects to your inner ear. And that's the majority of ear infections that we see. And this is quite common in children, of course. And so oftentimes if you have, I think, something like more than three or more ear infections before you're 2, they'll give you tubes. So they'll put a tube in your eardrum to allow drainage so that it doesn't build up back there and lead to another infection. All right, so now that's all the gram-positive stuff. Just a few slides on those just 2 gram-negative cocci bacteria, and that's going to be the Neisseria gonorrhea and the Neisseria meningitidis. Okay, and so these are going to be found in the mucous membranes of pretty much all warm-blooded animals. And so the 2 that are particularly pathogenic to humans are the ones I just mentioned, the Neisseria gonorrhea, which leads to gonorrhea, and the Neisseria meningitidis, which leads to meningitis. You got it. So their shape is often diplococci, kind of a bean shape. So when you're looking at it under the microscope, that's kind of the indication of what kind of bacteria the patient has. There are no flagella or spores, but they do have capsules and pili. Remember, the pili is used to transfer genetic content from one cell to another. They're strict parasites, so they don't survive long outside the host. They need to be in the host, and they're aerobic. Okay, so they like oxygen. Gonorrhea is an STD or an STI, and the things that lead to its pathogenicity are the fimbriae for attachment. Okay, they also have proteases that they can selectively target IgA, immunoglobulin A, which is the most abundant immunoglobulin in your body. And so, when trying to make antibodies to target and label those bacterial cells for attack or to surround them for opsonization, right, to render it basically unable to attach, it has these proteases that it secretes to break down our defenses. So gonorrhea is strictly a human pathogen. You can't get it from non-human animals. You get it through sexual intercourse with other humans. And it's one of the, not just really the top 5, but the top 3 STDs in the US. It has a relatively large infectious dose. If you guys remember IDs that we talked about at the beginning of the course, right? The number of cells or particles needed to mount an infection for gonorrhea, it's anywhere from 100 to 1,000 bacterial cells. And again, because it is a strict parasite, it's not going to be surviving very long on fomites, so on toilet seats and things like that. It's really hard to get it other than having sexual intercourse. So you can't blame it on anything else really, because the odds of getting it are pretty low. Now, there is such thing as extragenital gonococcal infections. So that's going to be not inside the genitals, so it's not going to be necessarily an STD. It's going to be largely in patients that could have more improved hygiene, and it ends up getting from the genitals to places like eyes, where it can be very serious. You get conjunctivitis, and so oftentimes the patient has gonorrhea from an STD, as derived from as an STD, and then it spreads to other places. Sometimes it can enter the bloodstream and from there go to things like your joints, which can lead to arthritis. It can be spread on the skin in some cases, and that will manifest as a rash. And then but that's not very common. And even less commonly will it go and spread to the meninges and the heart. Okay. But so this is an example of gonorrhea, primarily being an STI, but isn't always necessarily restricted to the genitals. Children can get it by having mothers who have gonorrhea. And so as they are being born, they acquire it through the birth canal. And that can lead to, again, a very serious conjunctivitis that can actually lead to blindness. So it can also inhabit the pharynx and the respiratory tracts of these infants that acquire it through birth. And so, like with group B strep, oftentimes this is screened for and then treated prophylactically um, to prevent passage onto the baby. Whoops. Now, Neisseria meningitidis, virulence factors, it has capsules, the Fimbriae. Again, it also has IgA protease, and it has endotoxins. So a little bit more than the gonorrhea. There are 12 different strains. Serotype A, B, and C are the ones that are found in most cases. Humans are the reservoir for this, and it's present in the nasopharynx of anywhere from 3 to 30% of the adult population. Those that are at high risk for this are going to be predominantly children, young adults, and those that are living in close quarters, like we all are now in the era of COVID. But what happens is that it enters the bloodstream and is one of those pathogens that can cross the blood-brain barrier. And once it does, it can grow on the meninges, which the meninges are present in the outer coating around on the outs between the skull, but on the outside of the brain, and enters there and can grow in the cerebral spinal fluid. And so it leads to a very fast onset of infection and leads to a very, very, very bad infection can lead to permanent neurological damage as a result. The endotoxins. What are you doing, buddy? Sorry, guys. No, what have you done here? The endotoxins. Blah, blah, blah. Bear with me. The endotoxins from this meningitis has severe epidermal lesions called petechia that develop, as you can see in this image down here. And that's going to be in about half of all the cases of Neisseria meningitidis. And you see like these really terrible lesions. They just look awful. Okay. And so it's basically necrosis of the tissue. And so very, very damaging, not only to the skin, but the neurological conditions of the patient. So something that is we try to prevent as much as possible. Okay. And then we have the gram-negative cocci Branhamella, which is also in the nasopharynx, is an opportunistic infection in patients with cancer or diabetes, or also patients that are experiencing alcoholism, which is going to wreak havoc globally on their bodies. And so their immune system, their kidneys, your liver, they can't fight off these infections. And then Moraxella is going to be found in the mucosal membranes. And then Acinetobacter, we've talked about Acinetobacter several times before. It's present in soil, it's present in our gut, but it can also be a pathogen if it localizes to the wrong tissues. Okay? Any questions on that?
Yeah, Dr. Franz, probably a silly question. I've been out of the STD world for some time, at least in my mind I have. What are the top two? You have gonorrhea is number 3. What are the top two?
Yeah, so I believe syphilis is one, and hepatitis B, but I could be wrong.
So on my research project, I'm looking at the yeast that has trichmosis. It's actually considered the number one sexually transmitted disease. But
Really?
it's not really talked about. Yeah.
Huh. Um, you know, they do tend to change depending on, you know, what, so different populations will see an uprising, and trichomoniasis, that could be. Um, chlamydia is also one that's oftentimes in the top 5. So so then that would be, so yeah, why don't we look it up? Anybody want to Google that really quickly? What are the top 5? And we'll go from there. I would think it's gonorrhea, syphilis, and chlamydia or hepatitis, but hepatitis B, but I could be wrong.
After this class, I start to question how many unknown STDs are out there as well.
Yeah. Yeah. So, you know, the thing about STDs is that it can often be a, you know, it's when we talk about the spread of diseases and stuff like that, it's often human, because it's STD, it's human behavior related, right? And so the spread is from certain behaviors in the population, whether or not they have access to clinical treatment. Okay, and you know, just their belief system. A lot of places where these STDs are present are going to be in places where, sorry, I'm a little distracted here. Hey, where people either don't have good access to treatment or don't have a lot of trust in medical facilities. And so we can see these numbers rise and fall. And so it's like certain regions will be like the number one, you know, city has the highest incidence of, say, gonorrhea, but then that can change from year to year. So I got some chats here. Maybe someone.
I'm also thinking of the unknown, right? Not just the ones that aren't treated because of you know, lack of accessibility, but stuff that's there that we just haven't discovered yet.
Yeah, I mean, that's possible. Certainly that things have been undetected, right? And particularly with viruses, that can be a profound under the radar kind of thing that circulates. And of course, that was HIV, right, back in the 80s. It's an STD that was circulating for a while before people isolated the causative agent. So definitely. Did anybody find the top 5 by chance?
I put a link on the chat.
Oh, okay. Well,
Gives
what were they, out of curiosity?
So this one was updated October 25th, 2020.
Okay, so
And
that's
so it's HPV is number 1,
mhm.
chlamydia is number 2, and trichomoniasis is number 3.
Okay. Interesting.
Gonorrhea is number 4.
Number 4. Okay. Yeah, so it kind of again changes from year to year on what's circulating more, but oftentimes HPV is pretty common as well. I didn't think about that one. Okay, what? Oh, did he eat your food? Yeah. Okay. Well, guys, any other questions on this chapter before we move on? Okay. Well, you let him eat your food. All right. So now I wanted to shift gears. That was gram-positive and gram-negative cocci. Now this chapter is on gram-positive bacilli. And then the next chapter, which we'll have to maybe get into Friday, will be gram-negative, because I don't think we'll be able to finish this in the next 15 minutes. So for the quiz on Friday at noon, it will be everything we covered on Monday and however far we get today. So there are 3 basic groups when we talk about the gram-positive bacilli. And that's going to be whether or not they have endospores and whether or not they have irregular shaped cells. And then we can further classify them down into their staining properties. So some of them we can't stain with the gram stain, so we'll use an acid fast stain instead. This is a diagram breaking down the different types that we're going to talk about and which category they come into. Hey. So again, all of these are gram-positive rods or bacilli, but then we have the endospore formers. So that's going to be bacillus and clostridium. Just one second, guys. Lucas. Okay, sorry about that, guys. So with endospore formers, you have aerobic or facultative anaerobes, and then obligate anaerobes. And so it's good to know these differences. So study this diagram and know that bacillus is in that category of aerobic or facultative anaerobe, and that clostridium is obligate anaerobe. And then we have the non-endospore formers. And so the regular shaped cell, regularly shaped rod, is going to be listeria. And then all the others are going to be irregular shaped. And so for that, we have non-acid fast, so they don't stain with the acid fast staining protocol. And that's going to be Corneobacterium and Propionibacterium. And then the one that does stain acid fast is Mycobacterium. And then we have this other category that is filamentous branching cell. So again, it's irregular shaped, and it's going to be Actinomyces and Nocardia. Okay. So this is the overall landscape of these gram-positive rods. And then from here, we're going to go into more resolution and talk about the different ones specifically. Bacillus anthracis, or anthrax, is going to have a central endospore. So a spore kind of on the inside, surrounded by this rod. And then if it doesn't have the spore, it's just going to be a vegetative cell. And it forms these long chains. So this would be kind of like a streptobacillus, right? It's a strip of rods. Whereas Clostridium botulinum, which we all are familiar with Botox, it's going to have a terminal spore that swells in what's called a sporangium. And we'll talk more about sporangia when we get into fungi after Thanksgiving break. But forms this tennis racket morphology. So if you look at the shape here, instead of having this just central endospore in the middle, like Bacillus anthracis, it has it at like a polar end of the cell. And that leads to like a flaring out at that end and gives it a tennis racket shape. Okay. So good thing to know, maybe be able to distinguish, if you see a picture of one of these, know which is which. There we go. The majority of these endospore forming bacilli are going to be saprobic. All right. Remember what saprobic is? they break stuff down. They secrete enzymes. And so they're going to be aerobic in nature. They're going to be catalase positive. That's just a test that we do diagnostically. You're either catalase negative or catalase positive, coagulase negative, coagulase positive. If we were having face-to-face lab at the end of the quarter, there's always a unknown bacteria that you would get, and then you'd run it through the series of tests that we do throughout the quarter: gram stain, acid fast stain, do these different catalase and coagulase tests to identify your bacteria. Unfortunately, that all it's thrown out for this quarter, but it's an important way to diagnose the specific bacteria that a patient is presenting with. And because they're saprobic or saprophytic, they have these enzymes that can degrade macromolecules in the environment because they're primarily in soil, and extract carbon from those macromolecules. And the ones of particular importance to humans medically are Bacillus anthracis, again, anthrax, and then Bacillus cereus or B. cereus, which is a common foodborne illness. B. anthracis is going to be, again, the picture that we showed earlier. It's got this endospore, non-motile rod, so no flagella, and a central spore in the middle. The virulence factors are going to be a polypeptide capsule, endotoxins. The majority of anthrax are in Africa, parts of Asia, and the Middle East. And there are three major types. So there's cutaneous anthrax, where spores enter via the skin. And this is going to be the least dangerous of the type of infection you can get. The one that's much more deleterious is pulmonary anthrax, and so that's what is a concern when someone opens an envelope in their mail and there are this white powdery substance in there. If you inhale the spores, then you get pulmonary anthrax. Very rarely there can be ingestion of the spores that make it to the gastrointestinal tract, and if that happens, it's pretty lethal. Okay. And I think that can also happen from bioterrorism as well, with getting it from soil or from animals. It's hard to get enough of it concentrated to get an infectious dose in the gastrointestinal tract, but it's not impossible. If someone has anthrax, they are usually given clindamycin, doxycycline, or ciprofloxacin. And usually that's done in combination with monoclonal antibodies to specifically target the exotoxins that then prevent cell entry. There are also vaccines that I don't think any civilians get vaccinated. It's usually more people in the military that have to travel abroad, or if you are in some kind of high risk job where you're traveling a lot to other places, and that's going to be a vaccine with live spores or the toxoid, and that's given to livestock. Whereas with humans, it's going to be the toxoid, and it's going to be over a period of a year and a half or so with annual boosters. Okay, so it's pretty intense regimen, and again, not something that is provided commonly to the public. B. cereus is a facultative anaerobe. It is modal, unlike anthrax. And so you can see all of these flagella on the outside. What kind of flagella pattern is that?
Perry something.
Peritrichus, yes. It's beta hemolytic. It can form spores. Its name, cereus, is Latin for waxy. Okay. And so, and that's in reference to the colonies when you grow it on blood agar. We grow it on blood agar because it's beta hemolytic and it likes blood. And so the colonies have a waxy kind of appearance to them, hence the name. It's commonly airborne. It's found in dust. And usually it's hard to disinfect or treat antiseptically for this because they have that thick spore. It can be in food because, again, this is the foodborne illness, and is tolerant to cooking. So tolerant to heat, tolerant to reheating, right? So when you have leftovers, it's a good idea to cook those leftovers, not just take them out and eat them cold, right? Because even if you reheat them, some foodborne illness like B. cereus can still be lurking. And this leads to nausea, diarrhea, abdominal cramping, vomiting. It's usually a fast, it comes on fast and it goes fast. So within 24 hours, symptoms should start to wane, which is nice because there's no real good treatment for it. You just got to let the body pass it through. However, patients who are immunosuppressed are going to deal with this at much higher rates. Again, because they're immunosuppressed, they're just subject to all kinds of different higher incidence of microbial infections. Clostridium is going to be also spore forming, as I mentioned, with these tennis racket shapes of the spore at one end of the cell. It's anaerobic, catalase negative. Many different types of species, and they can manufacture certain organic acids and alcohols through fermentation. They produce exotoxins and are commonly found in wounds, but also can be a foodborne illness as well. And in this case, it's a food intoxication because of the exotoxins that it excretes. Clostridium perfringens is going to be a wound infection of the soft tissue, very nasty. You see here that it looks terrible, right? And so this is going to be spores in soil that get into the skin, but can also reach places like the intestine and the vagina, and surgical incisions, fractures, diabetic ulcers where the skin is just open, and any kind of puncture wound, it's going to be bad, right? So, think gangrene. That was a huge result of this infection back in the, particularly World War I and before Civil War. These things were where soldiers had battle wounds and were left on the field for prolonged periods of time in the mud, where this Clostridium is found, gets into the wound, and then you have to amputate because the tissue is just completely destroyed. Lots of virulence factors. So alpha toxin is used to blow up the red blood cells. They have collagenase to break down, and hyaluronidase to break down connective tissues, and also prevent the phagocytosis, and then DNase to degrade the DNA, the genomes of the immune cells, trying to take them out. To prevent or treat gangrene, you got to clean the wounds. You usually have to debride the tissue, which is just like to just scrape it off, remove that infected tissue, which is quite painful. Heavy doses of cephalosporin and penicillin, and in extreme cases, a hyperbaric oxygen therapy. Okay. Now, why would that be important? Did I say here? I don't know if I mentioned that. Yeah, right here. So Clostridium is anaerobic, right? Doesn't like oxygen. So if you put it in a hyperbaric oxygen chamber, that can sometimes help eradicate the surface bacteria. All right, 10:53. Let's take a break and come back at 11, because I do want to go over a little bit about the BLAST lab. And I think if I can do a little bit more lecture on this chapter to get us further along. So let's take about a 7-minute break, come back at 11, and we'll do some more chatting, and then I'll let you guys go. Okay. Tetanus, also Clostridium, and this targets the muscular system. So it is a neuromuscular disorder, and often referred to as lockjaw in the early days, because that was one of the very telltale signs of this infection. Your muscles tighten up, and you can't open your mouth. This is like many of these bacterial species we've been talking about, is commonly in the soil. It can also be in the GI tracts of animals, where it doesn't seem to cause these neuromuscular disorders. They have spores that enter through puncture wounds, right? So you step on a rusty nail, you get inoculation with this bacteria. Burns, other kinds of things like trauma to the body, where you get crushed. Again, really just anything that will break open that epithelial barrier that prevents most of these nasty invaders from entering. The vegetative cells are going to be in an anaerobic environment, and they're going to secrete certain toxins that then lead to this neuromuscular disease. Tetanospasmin is that toxin, and so that leads to this paralysis, because what it does is the toxin attacks the motor neurons. So right here, that's all I got, buddy. So this is a picture here of the toxin, these blue dots, and they're going to essentially get in the way of this spinal neuron, and going to lead to just the blocking of that release of a neurotransmitter that signals for muscle contraction. And so what happens is the muscles just contract, and they don't get the signal to release. And so you end up just being in this state where you're just rigid and you can't move. It's very uncomfortable, very unpleasant. And typically this leads to death, because your diaphragm, which is a muscle, right, can't contract and relax to breathe, and so you suffocate. Luckily, there's a vaccine for this, and so it's not nearly as much of an issue nowadays, but it can be very bad. Clostridium difficile, or C. diff. This is a pretty pretty common disease, and is one of those few diseases that the use of probiotics seems to have a benefit. It attacks the colon, and what you're seeing here on in the image above, let me move myself. Here's a normal, healthy colon, and with C. diff, you get these large plaques of bacterial infection, just like riddled throughout the colon, and it's going to release these enterotoxins that damage the tissue present and leads to diarrhea and malabsorption of nutrients. It's a pretty common causative agent of diarrhea. And that's really, I mean, that's all we'll talk about. There's a lot that could be lectured on with C. diff, because it is compared to some of these other ones we've talked about, it's a pretty prevalent disease, and it can be hard to treat. But in the effort of covering all these things, we'll move on to Clostridium botulinum, or botulism. Again, when food is inadequately preserved or cooked, then you can have this spore-forming anaerobe present in the soil and in the water that contaminates the food and the beverages that we consume. And what happens is that it produces this botulinum toxin, okay? And the botulinum toxin also goes to the neuromuscular system and blocks the release of acetylcholine, which is necessary for muscle contraction. So similar to the one we just talked about here, tetanus. And this is why it's used, actually, Botox is used as a, if you get Botox by ingesting underprepared food, that's bad. But some people will take this isolated toxin and inject it into their face to, I guess, reverse the signs of aging. And what that's doing is it's essentially messing with the communication in your musculature and giving it like a kind of a tightened look, because the cells are just, stuck. The muscle cells are stuck. So if you're not purposely injecting yourself with Botox, but you're acquiring it from the food, some of these signs and symptoms of infection are difficulty seeing, blurred vision, swallowing, because that does take a coordinated musculature effort to swallow food and drink, and then certain neuromuscular symptoms, just like issues moving, right? Issues using your body. If you get it, oftentimes there'll be an antitoxin that is administered to patients, and that is just to prevent the more serious side effects, being cardiac or respiratory symptoms. So again, if your heart can't pump and your diaphragm can't contract and then release, then you're not going to be able to breathe. You're not going to be able to pump blood, so that you can die pretty quickly. You can also be treated with penicillin, And so, the best way to prevent botulism is to just properly prepare foods, right? Whether it's through canning, the canning process, adding preservatives and things like that. Um, Clostridial gastroenteritis. This is caused by Clostridium perfringens, which you'll remember we talked about a few slides back, which is gangrene. And right here. So if it's on the surface of the tissue, then that leads to this gangrenous breakdown. But if it does go into, if it's consumed and enters your GI tract through food that has not been cooked properly, then it can germinate and multiply in your GI tract, and the toxin that's secreted will attack the epithelial cells in your GI tract, and it's going to lead to abdominal discomfort, diarrhea, nausea. Luckily, it has a very different effect on the inside of you. It doesn't turn your insides gangrene, and it's flushed out within usually 24 hours. The nice thing about a lot of foodborne illnesses is that they come on quickly, and then they pass quickly. Okay. So that was the spore forming bacilli. I think we'll stop here for the lecture, and we'll take up non-spore forming on Friday. So for the quiz on Friday, you'll need to know about the gram-positive and gram-negative cocci, and then you'll need to be familiar with the gram-positive bacilli of the spore-forming flavors, okay? And then the rest of this will not be on the quiz. Hey, why don't you plug in? There we go. So any questions on that, on the spore-forming ones? Okay. What I wanted to do now is just briefly go into the lab to give you an idea of what it's about. And then you guys will be done for the day. Okay. So let me find that, see where that is. Okay. So if you go, let me share my screen again. If you go into assignments and you go to lab, there will be BLAST. Okay. Click on that, and it gives you this website link. Click on the link, and this is the lab that we'll be doing. So if you want, you can, there's different resources to kind of familiarize yourself with BLAST. Here's a slideshow that you can click on, and it will give you like a BLAST for beginners basics training, which you can do. But essentially, let's see what you do. Just click on NCBI homepage. This is what it looks like when you first go to BLAST. Okay. And all these different tools that you can check on. There's also PubMed if you want to look for papers that are published. But BLAST, they break it down into nucleotide sequences, whole genome sequences, single nucleotide polymorphisms, genes, proteins, and even a whole line of different chemical molecules. And so it's quite thorough, really handy database. And so what you're going to do is you're going to select nucleotide BLAST under the BLAST category. So let's go here, nucleotide blast. Let's go back. Nucleotide blast. And then it says input your nucleotides, ATGC, that fill one complete line into the search box. Okay, this is the query. So, oops. Let's see here. Nucleotide. That's not right. That's not what I'm looking for. Let's collection. Where is it? Where's the query? Blast. Nucleotide blast. There we go. Okay. So let me do that again for you guys so you can see how I did that. Rather than just go to nucleotide here, I think you should click on blast. And then this brings up just the whole blast search tool. And then from there, go to nucleotide blast. And so here, this is your query right here. This is going to be where you put in your sequence. And you can do several different things. Once you put your sequence in, it's important to select different criteria, standard databases. If you want a ribosomal RNA database, beta coronavirus, that's new. I haven't, they've just popped that one up recently this year due to SARS, but you can then say, I just want to search for things that are highly similar, or if you want to have it less identical, you can do more dissimilar or just somewhat similar. What that means is like anything that has a nucleotide sequence that is somewhat aligns, it will pull that up. Okay. Click on others under choose search set. That's going to be important because where is that? That's interesting that they don't have others. It's like they've changed the database a little bit, but I don't see others here. There we go. I guess they've changed it to 16S ribosomal RNA. If you have standard databases, it will just start looking at human databases, like human genomes, mouse genomes, because of mouse models and stuff like that. So if you click on this one, that will bring up bacteria, and that's what you'll want to do to search for this. And then what else does it say here? Select blast at the end of the page, a new window appears. Wait for the zones. Okay, so here's our sequence. Let's copy that. And let's put it in here. Let's just paste it in there and click blast. And see what comes up. Time since submission. So it's thinking here, it's searching. Nothing seems to be coming up. Why is that? That's interesting. That's interesting. It's not pulling that up. Let's go back and let's just let's do a fake sequence like they were talking about. So I'm just going to just put in series of A, T's, G's. Okay. And then let's blast that and see. That's a small sequence, so it might actually hit something. It's taking a little time. I don't know why it's not really working that well, that quickly. Might have to see how this goes. My blast isn't working. All right. Well, guys, that's strange. I might have to play around with this a little bit before I get it working. Blast genome. It's just so strange that it was working fine the other day. Mouse, no. Try this. Well, that is anticlimactic, isn't it? Okay, well, guys, sorry about that. That's a terrible demonstration on how to use Blast. I don't know why it's not pulling anything up. Why don't you guys give it a try? And if you're able to get it to work on your end, maybe it's, I don't know, maybe it's my internet or something. Let me know. In the meantime, I'm going to try to troubleshoot this and see why it's not pulling up and maybe give you guys some different instructions. That's very strange. So give it a try on your end if you have some time now. Let me know if you have the same problem as I am, and I'll see if I can find a workaround, see why it's not working. Maybe I've forgot to tweak some little criteria here. Okay. Any questions on that? I know that was awful, but what are we going to do, right? What can we do? Like I said, play around with it. Let me know if you're able to get any success. And if not, then I don't know. Unfortunately, we may have to scrap it. I hope not. Yeah, what a blast, huh? Lame. Okay. Any other questions while I have you guys here about anything?
I have a question. I think you mentioned you were posting the homework. Was it this week or next week?
I'll post the homework today, and it'll be due next week.
Oh, okay, got it. Thank you.
I haven't posted it yet.
Dr. Franz, can you talk a little bit about the newest, latest, greatest going on with COVID, SARS-2?
Yeah, happy to. So there's actually a couple neat papers. One paper came out showing that there is pretty good antibody recognition and production that's long lasting. So one of the issues that scientists were concerned about was that the possibility of reinfection because you're not getting the proper immune response and subsequent antibody production for lasting immunity. And so there was a paper just came out in Nature Microbiology that I saw this morning or maybe last night that was suggesting that that's actually um, that it's a neutralizing antibody that is found in patients that have convalesced, and so they look at their convalescent serum, and it's long lasting. So that bodes well for vaccine production, and I'm sure you guys probably know if you've been tracking the news is that Pfizer and Moderna are both in a hot competition right now with their vaccines that are so. Pfizer a few days ago was reporting better than 90% uh, safe efficacy. And then Moderna came out and said, well, we're like almost 95%. And it's kind of like, the space race and the arms race with the Soviet Union back in the 60s. It's like now Pfizer says, oh, never mind. We're also 95%. So it'll be really interesting to see how these two vaccines play out and which one ends up um, being the one widely used if, unless someone else comes up with something or, it's possible that even with 95%, that we could see side effects crop up in one of these studies, but it's looking very promising. I believe the Pfizer vaccine has to be stored at a very low temperature, which makes distribution of that one um, more uh, technically challenging, whereas the Moderna vaccine is doesn't have to be at that cryogenic temperature. Um, so yeah, it's interesting there. What else? Any other questions that you were thinking? Was that what you were looking for? Or was there something else you were curious about?
No, that's great. That's the
Yeah.
information that I've been reading and listened to on the NPR.
Yeah.
Um, I, I guess there's still a little bit of confusion on for me and I just need to read up on it more. The mRNA, what happens there on that uh, molecular level?
Mhm.
Um,
So you mean the mRNA used to make the vaccine or
yes.
yeah, yeah. So I believe both of these are targeting the spike proteins on the surface because the spike proteins of the viral particle is what binds to the ACE2 receptor. And since it's on the outside, it's the easiest to form antibodies to target those spike proteins. And so um, that's what they're using. They're using sequences of they're using the genetic sequence that makes the spike proteins. It gets translated into the proteins as a source to produce antibodies for the immune response.
Thank you.
Yeah. Exciting times, I mean, of course, when a vaccine does come out, and I was very skeptical, earlier this year when people were talking about the possibility of a vaccine this fall, um, because vaccine development does take typically a very long time. And clearly researchers were under a lot more pressure to come up with a vaccine. And so you might be working around the clock, whereas with trying to find a vaccine for dengue virus, scientists aren't working around the clock. They're working their normal hours, and so that can speed it up a little bit. But you're at the mercy of just the time it takes for experiments to progress, and the time it takes to accumulate enough data to show that these vaccines aren't going to be detrimental and that they're unsafe. And when you're talking about a vaccine, which to a fair cross section of people, the word vaccine triggers a response of barbarism and terror. And to rush something is not going to instill confidence. in the population, right? And so the faster you have a vaccine pushed out there, the the chances of people not wanting to take it are higher. They don't want to be the guinea pigs, right? Even if you do have science that says it's safe and effective, it's going to be challenging to get enough people to take that vaccine to have a reasonable, herd immunity. And so um, we'll see how long it takes for one of these vaccines to be put into use, and then from there, how many people will take it, who will get it first, right? Healthcare workers, immunocompromised, elderly will probably be the first ones to get it. But to roll out this vaccine is going to take a long time, right? And now we have an administration change with the government, and there's all kinds of, um, you know, um, there's at least right now there's a lack of communication, and that's going to delay things, right? And so, unlike the movies where there's an outbreak, a pandemic, and then they come up with the antidote or the vaccine, and then life is back to normal. It doesn't work that way in the real world. That's a Hollywood phenomenon. And so, we'll be dealing with this probably for quite a while.
I'm just thinking the other spot of confusion for me is they with the initial discovery of this vaccine, they're now able to bypass uh, a ton of work and then get right to the goods. I'm generalizing in a bad way.
Well, I think that
Clarify that.
what happens is you can work on the previous findings. If you're using the same input, for instance, then there are certain biological principles that can be applied, and so you don't have to necessarily, you can, I guess, jumpstart it a little bit. You don't have to take it through every single test. But I don't really know a whole lot about how they're making these vaccines. I think that there's not really anything published yet. Right? And so you can't read, all you can read is press releases and news articles, right? And a press release and a news article it's not going to be scientifically meaty. And so I don't know if that's due to just, it takes time to publish, or if the competitive nature of things, they want to give out their secret sauce, so to speak. But there just hasn't been a whole lot that I've come across that details the science that's going on with the development of these.
Yeah, please share the Nature article that you read last night regarding I think it's like a 6-month immunity or something like this was what the news was boasting.
Yeah, well, I mean, this is, so there's been all kinds of conflicting, things in the news, and people have been getting it, being infected a second time and dying from it, and how long is immunity going to last? Is it lasting or not? This paper is suggesting a longer lasting immunity, which is good. Very, very optimistic. I'll share that for sure. I'll put it up. Anything else, guys, about COVID-19 or the class or anything while we're here? Okay.
Professor, I have a question for the
Yeah.
quiz. Will it be covered for the lecture last Friday or not?
Not for the quiz. Oh, yeah. What was that? That was.
That's what the bacterial agents.
Oh, yes, that will be. Yeah, that will be fair game on the quiz as well. None of the microbiome or hygiene hypothesis material will be on the quiz, but the microbial agents, that was talking about the spirochetes and stuff like that. That is also potentially on the quiz.
Thank you.
Thanks for bringing that up.
Yeah, thank you.
All right, so I'm looking at my blast and I see that no significant similarities were found for that sequence that I put in there. So I'm just gonna have to, oh, that's because that was the fake one that I put in. I see. Well, I'm gonna keep trying, guys, with this blast. I'd really like it to work for you. But if not, we will adapt and overcome. So I'll play around with it. I'll keep you guys posted on that, on any progress or lack of progress. And again, the kits are ready to pick up. Friday, we'll have our morning on campus virtual lab, and look out for the homework to be posted soon. And we're almost there. I'll keep an eye out for your proposals on Friday, and just hang in there in the meantime. Okay. Well, you're free to leave if you want to stick on and ask me any other questions or anything. I'm here. But otherwise, I'll see you guys Friday morning.